This tutorial introduces the HEXAGON-IMAGING-PAM and places it within the broader context of our Walz imaging-PAM family. The topics presented here highlight selected and typical applications. For a complete and detailed description of all instrument functions, please refer to the manuals available on the Walz website. Chlorophyll fluorescence is one of the most widely used techniques for studying photosynthesis. One key advantage is its high sensitivity. Fluorescence signals can be detected at very low pigment concentrations, enabling reliable measurements to be taken even when working with small amounts of material. Equally important is the versatility of fluorescence measurements. For example, different types of information can be obtained by using different time domains, and several physiological processes modulate the fluorescence signal. Another significant benefit is that chlorophyll fluorescence measurements are non-invasive and relatively straightforward to perform, making them ideal for comparative studies. There are two main ways in which chlorophyll fluorescence imaging can be used. Firstly, it can be used as a standard fluorescence technique, whereby many point measurements are performed simultaneously. This essentially produces a set of bulk measurements acquired in parallel. Secondly, imaging enables the investigation of sample heterogeneity. Spatial differences within leaves or tissues can be detected during processes such as stress development or disease progression. The key concepts in this context are therefore spatial and temporal heterogeneity, bulk measurements, and comparative analysis. Walz is a leading manufacturer of highly sophisticated chlorophyll fluorometers. We offer a range of imaging solutions for various applications and resolutions. The following section will familiarize you with our different imaging-PAM instruments. The imaging-PAM mini version measures a relatively small area, but offers high spatial resolution. The instrument is portable and can be brought directly to the sample, enabling us to image in the field. The imaging-PAM microscopy version is designed for chlorophyll fluorescence imaging at the cellular level. It can be used to examine individual cells and chloroplasts. In conjunction with the modified fluorescence microscope, it offers the highest sensitivity of the imaging systems. Chlorophyll fluorescence imaging was originally developed to visualize spatial heterogeneity in plants or leaf tissue. As imaging systems grew in size and power, the ability to measure multiple samples simultaneously became increasingly important. The imaging-PAM Maxi version covers an area of 10 × 13 centimeters. This allows for the simultaneous measurement of either several leaves or an entire 96-well plate. The HEXAGON-IMAGING-PAM further increases the imaging area by a factor of 4, resulting in an area of 20 by 24 centimeters. For example, this allows several Arabidopsis plants or 4 96-well plates to be measured simultaneously. Several technical requirements must be met for reliable chlorophyll imaging measurements. One critical aspect is the homogeneity of the illumination. A homogeneously illuminated area is crucial for high-quality measurements because inhomogeneous illumination makes it difficult to distinguish between light intensity effects and true physiological differences. Simply distributing LEDs evenly across a panel would result in higher light intensity in the center and lower intensity at the edges. Therefore, for the HEXAGON-IMAGING-PAM, a modular illumination system based on hexagonal units has been developed. It uses a deliberately non-homogeneous distribution of blue LEDs. In particular, the LEDs are arranged in a statistical pattern so that their light cones overlap as evenly as possible. The HEXAGON-IMAGING-PAM uses blue LEDs, which provide a high output. These LEDs give a strong signal, which lets us detect chlorophyll fluorescence very sensitively. Additionally, the HEXAGON-IMAGING-PAM uses fans for active cooling, which helps minimize the effects of heating. After discussing the technical features of the imaging-PAM systems, we now move to the software that connects measurement and analysis. The Imaging Wind software provides complete control of the instrument and displays fluorescence parameters in real time during the experiment. At the same time, It offers a range of tools to visualize, organize, and analyze the data collected by the system. The system offers predefined protocols, such as rapid light curves or induction curves, which are easy to use and cover many standard applications. At the same time, you can fully customize your experiments using scripting, allowing you to program completely user-defined measurement sequences. In the following sections, we will take a closer look at the main functions of the software and how they support efficient data analysis. When using an imaging PAM instrument, chlorophyll fluorescence is measured for every pixel of the camera image. Each pixel therefore represents a small measurement point on the sample, allowing spatially resolved analysis of photosynthesis. During the measurement, the instrument continuously records the steady-state fluorescence signal FT for every pixel. When a saturating pulse is applied, all PS2 reaction centers close temporarily and the system records the maximum fluorescence FM'. From these two values, parameters such as yield 2 or non-photochemical quenching can be calculated. Because thousands of pixels are measured at once, it would not be practical to display numerical values for each point. Instead, the software converts the calculated values into a false color image, where each color represents a specific parameter range. A color scale next to the image shows how colors correspond to numerical values. This makes it easy to quickly identify spatial differences in photosynthetic activity across the sample. One of the most important analysis tools provided by the software is the area of interest, or AOI for short. The IMAGING-PAM Maxi version allows up to 100 areas of interest to be defined, which is sufficient for a single 96-well plate. The HEXAGON-IMAGING-PAM enables the definition of up to 400 areas of interest. This allows the simultaneous analysis of up to 4 96-well plates. Areas of interest can be circular, rectangular, or polygonal, and even entire leaves can be defined. The software handles averaged AOI values and individual pixel data differently. Specifically, the average fluorescence value of an AI is recorded continuously. This produces classical induction curves, for example. The software allows different parameters to be plotted together for the areas of interest and evaluated live during the measurement, providing immediate insights. All individual pixel data is captured during saturation pulses when the F and FM images are recorded. For full image analysis, these data can be viewed as a dynamic slideshow, with successive saturation pulse images displayed one by one, each showing a single parameter. This makes it easy and intuitive to track changes across the sample. The mean over area of interest option can be activated or deactivated in the software. When enabled, pixels with a value of 0 are included in the average, which can result in lower mean values. When disabled, these 0 values are ignored. The appropriate approach depends on the biological question. For example, necrotic areas can be included or excluded from the average, depending on the desired analysis. The IMAGING-PAM Maxi and HEXAGON-IMAGING-PAM can also be used to measure suspension samples in standard 96-well plates. In the software, areas of interest can be defined to correspond to the individual wells. Well plates are available in different colors. Black plates help reduce reflected light, and additional filter plates can be used to further minimize reflections and improve measurement quality. A unique application of the IMAGING-PAM Mini version is the detection of GFP fluorescence in addition to chlorophyll fluorescence. This capability allows researchers to combine measurements of photosynthetic performance with the visualization of fluorescent protein markers in the same sample. The system enables convenient switching between filter sets for chlorophyll fluorescence and GFP detection, making it possible to acquire both types of information within a single experimental setup. Due to the high spatial resolution of the Mini version, small structures and spatial patterns of fluorescence can be visualized in great detail. This makes the system particularly suitable for studies requiring precise localization of fluorescence signals within tissues or small samples. At last, applications from the literature illustrate how Walz imaging-PAM systems can be used in research. In earlier studies, for example, chlorophyll fluorescence imaging was used to visualize how the herbicide DCMU spreads across a leaf over time. Changes in photosynthetic activity could be followed directly as the inhibitor moved through the tissue. Imaging can also be used to study other physiological treatments. In this example, a weak acid is applied to leaves, which artificially acidifies the thylakoid lumen and thereby alters NPQ formation. The fluorescence images show that after this treatment the leaves behave similarly to the NPQ4 mutant, which is known to be impaired in NPQ formation. This illustrates how fluorescence imaging can visualize physiological responses across the leaf surface. Another example focuses on studies that have investigated the effects of temperature on photosynthesis using fluorescence imaging. In recent years, several publications have used imaging systems to analyze temperature responses in suspensions and leaves. For leaf measurements, the leaves can be placed on a Peltier-controlled plate, which enables them to be gradually heated or cooled during the experiment. When leaves are gradually heated, damage to the manganese cluster of photosystem 2 is associated with an increase in the F0 value. The inflection point of this increase is defined as the critical temperature, above which significant damage to the manganese cluster begins to occur. A similar increase in F0 can also be observed when the temperature is lowered below 0 degrees Celsius. In this case, however, the manganese cluster is not damaged. Instead, at very low temperatures, the electron transfer from QA to QB on the acceptor side of photosystem 2 significantly slows down or stops. Overall, such experiments help to estimate the critical temperature thresholds of photosynthetic performance, and these changes can be clearly visualized using chlorophyll fluorescence imaging. These examples illustrate different experimental approaches and applications using the imaging-PAM systems. A regularly updated collection of scientific articles demonstrating the use of our imaging instruments is available on the Walz website. These examples highlight different experimental approaches and may serve as inspiration for your own measurements. Thank you for listening to our introduction to the imaging-PAM instruments. For more information, please visit our website and explore our other online tutorials.